Okay, let's get back to the log tree issue. The performance. So I talked about this hierarchy. I said on the highest level of the hierarchy we distinguish between proteins that are so-called water-soluble and transmembrane. So they go through the membrane. That turns out to be the simplest decision, and a method that existed before sort of get 95% of the classification right. Anybody in the room? This is a kind of question that I would typically not ask, but do you remember what may be the reason at this point for making mistakes in that decision? This is typically a question that I would call too biological for you guys. Do you still remember what, in this particular decision that goes right 95% of the times by the best tool in that sort of exists, or one of the best tools, what's the challenge in this decision for the biological side of the problem? So we're looking at proteins that have a membrane region and proteins that don't have a membrane region. So one of the issues is that proteins that don't have a membrane region but get secreted into the secretory pathway have a signal peptide at the beginning of it, and this signal peptide has a hydrophobic region that looks like a membrane helix. So that is the major reason for this not being closer to 100%. So that's the major reason for this error here. Okay, and then we go further down in the pipeline, and prediction accuracy drops. And prediction accuracy drops because ultimately the problem it gets increasingly difficult. So that is sort of the biological view of it, because overall you distinguish two things that are very, very different. Proteins that have membrane helices at least have many regions in them, or are supposed to have many regions in them that are recognizably easily. While when you go further down here and you distinguish between plastids and chloroplasts, so these are different, you could argue different types of plastids, between something that is maintained in or retained in the Golgi and goes on into secretion. Maybe some of these decisions are complicated because the biology of the problem is such that the proteins are really similar. So the retention in Golgi, for instance, is something, again, this will not be asked, but just as an idea, this is a sort of signal peptide that sits on the other side, so on this so-called C-terminal end of the protein. And very, very few of these retention signals are actually known, in the sense that, like the nuclear localization signal, I can experimentally put that into any protein that will get imported into the nucleus. And these signals are not strong. They're sort of probabilistic signals. So there must be something else on top. And that means they are somehow looking more similar to the proteins that are not retained, right? So there is more detail to be known, and that is also making it difficult for the system. There is another thing that has, if you want, nothing to do with biology. So let me ask you, what do you guess if I asked, if I sort of sorted all the classes that have a low performance here, and I asked what is common to them, what do you believe will be common to them in terms of your data? In fact, this is not a question that is really about biology at all. It's more about machine learning. What do you believe could be a reason why we make mistakes in classifying samples? There are a couple of reasons that are not biology, but sort of generally when machine learning goes wrong. What are reasons? Several of you, yeah?
Noisy data.
Noisy data is one reason. Ultimately noisy data, again, what it means, just to repeat this, means that for the classification task, noisy data has, say you have two classifications, red and blue. Some are sort of labeled as blue that are actually red. So that is one way of calling noisy data. So class confusion is a sort of translation of the word noisy data. That could be one reason. What else?
The data is too complex to be covered by this model.
So ultimately this means that now you're saying something that actually is much more complex in its statement. So let me take that apart. First, what you're saying is that what I use as input features is not enough to distinguish my data. Okay? That's one thing. But there's something else. And that something else has to do with a mere number. Almost always in machine learning, whenever the classification is non-trivial, so you cannot make it by a linear classification, whenever you have sort of a real machine learning, then it works better the more data you have. It has to do with the complexity of the story, so this word of complex is sort of implicitly in there here. But there's something much simpler than the word complex that we can typically not sort of unravel, which is just number. So I do not have this slide here, but when we looked at the number of samples and performance, there was a very clear correlation. Okay? Again, number of samples is always not just the mere number, it's also the number that are different and that bring new information, so it's not that easy to measure that. But that's a general thing to remember. Now, then there's always this issue that you have to compare your new tool with many other tools based on similar sets. And then you get a slide as complex as this one here, although you use only one single criterion for success. Let's just look at this example here. So we are predicting, this method is called log-tree2, log-tree2, and that predicts subcellular localization in 18 states. Nevertheless, here I show a 9-state performance. Why do you believe this is on the slide? I have 18 states, why don't I give the 18-state number? Incidentally, the 18-state number is actually not that different, because 9 of those states are sort of minority states and they don't really change performance much. To a few counts, but that is not the reason why this is done. At some point, you always have to compare yourself to whatever exists. So this existing thing could be random, this existing thing could be experimental performance, and this existing thing could be an idea what whoever uses your tool will need. There's a different ways of comparing. And the simplest one to start with is you compare yourself on the same data set with an existing method. Now, it happened to be the case that the largest method, when we published this first, predicted 9 states. So we could only compare these two methods one-on-one based on those 9 states. Why this is a 9-state performance. Now, this is the existing method, and that is the new method. Is the new one better? Everybody clear about the answer not being? So the new one looks better, yes, but we cannot with any statistical significance claim that it actually is, because the standard error is 14 percentage points, and that ultimately comes from, in order to compare to this method, I may show you other numbers of which this number is much lower. For our data, the estimated number had a much smaller standard error, because we had a lot of data. But for the data for which we could compare our tool to another tool that existed, that was so high because that data set was so small, because we needed proteins that both of them had not used, or that were different from anything that those two had used. Okay. Here is a comparison in 5 states to an old tool that we had. So the difference between these two here now is this tool here essentially only has the same hierarchy, but it only uses neural networks. While this one, not only, it uses neural networks for that each neural network sees the composition of the entire protein. Variety of information content for the composition. This is the step up here to SVM, and the SVM using the profile kernel idea. Again, based on completely using new data, to be sure, we cannot say that it paid off. That is the problem with it's the killer of small data sets. So we believe, and the trend continues. So here's for bacteria, and so again, even here, where we have a difference between 86 and 72, even here, we cannot make a statistically significant statement. So we have a large margin between these two numbers, but the data set is so small that we cannot tell statistically significantly that this is better. Now, there are two different things. So you publish that something is statistically significantly better. Yeah?
But why? I don't understand why if there is 18 and there is 21, we cannot say that the one on the left is better.
Because, so again, we are starting, if you just took this number 86 and you subtracted from 86 one standard error, minus 18, 86, I'm sorry. So then we are getting at 68.
Okay.
68 is smaller than 72. And that is one standard error. One standard error essentially means we have roughly 33% of the population. So this is not even a strong significance, right? So this is a strong overlap of two distributions is what is underneath this. Now, you will never go out and say, well, I believe this is better. But you can also not publish that you show that this is better. You can also not show that this is worse. Now, what it ultimately means for your own research, are you now going, because you cannot establish that this is better, going to use, if you make the tool available for the world, well, statistically significant, you cannot distinguish, so are you going to use that one then? No, right? This is a case where the numbers are the difference is so large that you believe this clearly is better, but you cannot prove it. But I would still, if somebody gave you a prize for running the best method, so I give you 100 proteins and you have to run the best method, you will always pick that one. So those are two different realities of statistical significance, if you want. Now, you may remember that at some point I said I want a variety of features. I want to have a method that predicts for eukaryotes or prokaryotes. I have that. I want it in many, many states. I have that. I wanted it to beat all the state of the art, or beat at least as good as the state of the art. I have that. But one issue was, how sensitive is this against changing the sequence? So for instance, changing the sequence here is measured in three different ways. In one, I take the 31st residues out. Just remove and ask how good is my prediction method without those. I take the last 30 residues out, and here I randomly remove 1/3 of the entire protein, but one stretch. I'm not removing single residues. I take one stretch, that is 1/3 of the length, and I remove that. And I essentially ask, how does performance go down for a variety of methods? Here's the method that we have. So this is +-2 here now because we have a much larger data set. Because this comparison here is with respect to itself. So the comparison in this direction is not really relevant because we're using different data. And you see that yes, it goes down, clearly, for all of these. The worst is that you will pick some point and remove 1/3 of the protein. But if you compare it to other methods here, it does not lose as much. In particular, you see that the moment you remove the beginning of the protein where the signal peptide sits, typically these tools lose a lot. And again, that's the strength of this profile kernel, because you have the 8,000 dimensional vector or higher 1,000 dimensional vector. There are features throughout the length that are relevant. Not only the N-terminal signal. This is the decision of the SVM does not zoom into the only one strongest signal, which are the motifs at the beginning. Okay, that is what we clearly can show. We realized with that, although again, so picking this out here is a strong impact. But another part is the comparison here. So for other tools, you see that in fact picking the end term, the beginning of the protein, is the worst influence. Picking randomly 1/3 is not hitting them as much. While for that tool here, picking 1/3 is the worst you can do. And that typically is, so because the decision is based on a bunch of motifs or k-mers, and that removes 1/3 of all the k-mers. Most proteins are longer than 30 times 3, 90 residues. Most are longer. So this one here removes more than any of these. So this removes more of these motifs or the k-mers that the SVM makes the decision on. That is why for our tool this is sort of the strongest effect. And that makes a lot of sense. Okay, now comes the final question. So once you have this tool that does machine learning only, we have set it up deliberately such that you can do homology-based inference independently of that, and that you apply this tool only when homology-based inference doesn't work. I don't have to repeat homology-based inference to any of you. So how would you combine these two now? So now you have a de novo prediction method, and you have your homology-based inference tool. How to combine the two? Well, one is you could assume that the user will know. But this, in fact, is what we historically did, and it turned out to be not a clever decision. So now we sort of have begun to put it into the tool. And that's why this tool is now called Loctree3, essentially combines these other two tools. How would you do it? What's the simplest way of doing it? Yeah?
Well, you get probabilities out for the classes, and you can compare the homology-based and the log2 probability and just choose the higher one.
So that is, in fact, a very good idea. And it's not the solution that we took. But it should be better, and I don't know why we didn't do it. I cannot remember why we didn't do it. So the query and the experimentally known sequence similarity is above some threshold t. And then you know that the performance for t is p, t. So you have you know what the probability of being right in your prediction through homology-based inference for a given t is. So now in your case, essentially for your de novo, so the Loctree2, for q gets a prediction. And this prediction comes with a certain probability. And then you can simply ask, is this probability here higher than that? As long as that one is higher, you take it. That's one way of doing it. The problem is that it could be, so the problem ultimately is to sort of measure the probability of homology-based inference in a right way. These are, if you remember, fairly sigmoid distributions. So homology-based inference works, and suddenly fails. Turning that into a probability number is sort of challenging. So what do you do now? Typically, the way you do it, you should choose this point to say, okay, that's the threshold. I'm going to say homology-based inference works only above that threshold. Only for t's higher than that, homology-based inference works. Now, that means that if the curve looks like this, you have very little space here to sort of linearize in a probability function. That means that this p here is actually not really, possibly not easy to become meaningful in the sense of comparing it to that one. That's the downside to it. But I'm not sure how far we really explicitly try to combine them in a way. Right out of the box it doesn't work, that I know. But, so what else can you do? Well, it's sort of written there, right? The alternative is really the dumbest thing you can do, which is to say, if the t is above that certain threshold, you take homology-based inference. If not, you take de novo. Okay? So a simple, not a probability, but a simple threshold, and this is in fact the path we took. And so what you see here are different classes. Here's a geometric average between recall and precision. The blue one is just the homology-based inference for where it works. The red one is the de novo, and the green is the combination above a certain threshold. Anything that you observe? So the curve, let's just do the, again, and I can pause the camera for that anyway, I can switch off the camera from here on.